Industrialized society's increasing dependence on fossil fuels is a cause of great concern in a world with an increasing demand for non-renewable resources such as coal, natural gas, and oil. Biofuels are an alternative to these polluting resources, using biological material to produce cleaner, renewable fuel. Ethanol is a biofuel most commonly used as an additive to gasoline, but it has other applications such as in cooking fuels and gels. Because fossil fuels are quickly being depleted and the prices are high, the market is interested in using ethanol as a greener and potentially less expensive alternative. Ethanol is produced by the fermentation of sugars by yeast. Consequently, the biofuel industry is constantly trying to determine the optimal conditions for factors affecting fermentation rates. Thus, the purpose of our experiment was to determine whether or not the different sugars are more efficiently fermented by the Saccharomyces cerevisiae strand of yeast. It was hypothesized that different sugars would, indeed, produce different fermentation rates. Because ethanol and carbon dioxide are both byproducts of fermentation, the production rate of carbon dioxide was used as an indicator of the production rate of ethanol, because the gas production was quantifiable. To measure the production of carbon dioxide, 1 gram of sugar was weighed on a scale and dissolved in 10 milliliters of water. Afterwards, 10 milliliters of 8% yeast was added to the sugar-water mixture, and it was left to incubate for 5 minutes while occasionally stirring. Next, 3 milliliters of the yeast suspension was taken into a 5 milliliter syringe. Inverted, 1 milliliter of air was additionally drawn in, and the tip was secured to a 2 milliliter pipette with a drop of water centered at the 0 milliliter mark. Held upright, the height of the water droplet was recorded every 2 minutes for approximately 20 minutes following the apparatus's assembly, measuring the production of carbon dioxide gas in milliliters. In addition to a glucose control, 4 different sugars were used: fructose, sucrose, maltose, and galactose. 1 gram of each sugar was used with the same fixed quantities of water and yeast for every trial that was held. We held 2 trials for glucose, and the results were pooled with the entire class. Additionally, we held 5 trials for each of the remaining 4 sugars, and the results were pooled with Sophie Aubé's team from the 350 class. For each trial, a graph was produced, plotting the quantity of carbon dioxide gas in milliliters as a function of the time elapsed in minutes. To determine the carbon dioxide production rate for each trial, a slope of the linear portion of the graph was calculated. Next, an average carbon dioxide production rate was calculated for each sugar by averaging the 5 slopes of the different trials. Glucose had the highest carbon dioxide production rate at 0.11 milliliters per minute. Sucrose had the second highest rate at 0.1066 milliliters per minute, with fructose at a slightly lower rate of 0.10636 milliliters per minute. Maltose had an even smaller rate of 0.08342 milliliters per minute. Finally, galactose had the smallest average carbon dioxide production rate at 0.00156 milliliters per minute, which was significantly smaller than its 4 counterparts. Because glucose had the greatest carbon dioxide production rate, it can be concluded that it also had the greatest production rate of ethanol, making it the most efficient sugar for bioethanol production. It follows that sucrose is the second most efficient sugar, followed by fructose and maltose. Finally, galactose does not produce a significant amount of carbon dioxide, with a fermentation rate nearly 70 times slower than glucose. Thus, galactose would be the least efficient sugar to use in industrial production of ethanol. Based on the results, glucose has the highest fermentation rate by yeast. Therefore, it would be the most efficient sugar to use in the industrial production of ethanol because it is more quickly fermented and, consequently, produces greater quantities of ethanol in a given period of time. However, the controlled variables in the laboratory are not necessarily representative of industrial applications. Foremost, the various carbohydrates used in the laboratory experiment were pure sugars. In an industrial context, pure sugars are too expensive to use because the purification and refining processes involve additional expenses that the bioethanol industry can circumvent by using sugars directly derived from raw biomass sources, such as sugarcane and corn. Globally, Brazil and the United States are the biggest producers of fuel ethanol, primarily using sugarcane and corn as biomass sources respectively. On the one hand, sugarcane stems have a sucrose content of approximately 12 to 20%. Further, sugarcane has a high growth rate and is ready to harvest after 18 months, producing a fair amount of sugar for fermentation purposes. On the other hand, corn grain has a starch content of approximately 70.5% and is harvested from fields annually. Starch is a polysaccharide composed of chains of glucose molecules joined by alpha-1,4 linkages. Our results conclude that glucose is the most efficient sugar for bioethanol production, and, coupled with the fact that there is a greater quantity of glucose in corn than there is sucrose in sugarcane, it would appear that biofuel fermentation would be most efficient using corn as a biomass resource. Despite the fact that yeast can ferment glucose, it cannot readily ferment starch. Consequently, for corn to be used in bioethanol production, its starch content must first be saccharified, or broken down into its monosaccharide glucose components. This is a process that is not only time-consuming, but expensive. Finally, sugarcane occupies less field space than corn, so more sucrose can be harvested from sugarcane in a limited space. According to the European Biomass Industry Association, the estimated production cost of bioethanol by US corn is approximately 0.42 euros per liter, and the estimated production cost of bioethanol by Brazilian sugarcane is 0.16 to 0.28 euros per liter. Even if glucose is more quickly fermented, as demonstrated by our results, its fermentation rate is only 3.1% faster than sucrose. Therefore, on an economic basis, fermentation of sucrose is the best alternative because it is more economically efficient. Maltose and fructose are also quickly fermented, but they are found in smaller quantities in biomass sources, making it less accessible than glucose and sucrose. Finally, galactose, found in biomass sources such as sugar beets and dairy products, is extremely inefficient with regards to ethanol production, as demonstrated by our results. The bioethanol industry is moving towards the production of cellulosic ethanol, using cellulose found in lignocellulosic biomass as a source of glucose. There is a complex chain of enzyme-driven chemical reactions required to break the cellulose down into fermentable sugar, but research is being conducted to find a more efficient and less costly method to do so. After all, lignocellulosic biomass is the most abundantly available raw material on Earth, and since our experiment concludes that yeast ferments glucose the most quickly, finding an efficient way to break cellulose down into its glucose components would allow us to achieve a more efficient means of producing bioethanol, a safer, more sustainable, and more environmentally friendly alternative to fossil fuels.